Now we move on to the next presentation. One of the first people I called when we started organizing the conference was Dr. Blumberg, because he walks the line between science and advocacy and is willing to have his science applied in the public sector and the public forum to advance changes in policies. I don't have a long introduction for Dr. Blumberg, but I do want to push his book, which is out front, The Obesogen Effect, because he's done extensive research over his career on chemicals that are causing hormonal effects and effects that affect multi-generational impacts. So with that short introduction, Dr. Blumberg, welcome and thank you so much for what you do.
So thanks very much, Jay, for the nice introduction and for the invitation to come and speak here. Also thanks to Dean and the other speakers for introducing the general topic of how pesticides and chemicals in the environment are bad for our health. What I want to do today is talk about a specific example of something which you may or may not have heard about. So what I want to tell you is that the environment influences health and disease. You've heard that already. I want to say that there are such things as obesogens and that we believe they contribute to this obesity, it says here epidemic, but it's really a pandemic. It's worldwide. It's not limited to any one location. I want to show you that the effects of these exposures are heritable. So the results I'm going to briefly summarize actually go to this generation and the next generation. So in human terms, if this woman was exposed while she was pregnant, both her children, her grandchildren, her great-grandchildren, and her great-great-grandchildren will show an effect. And that's something we've never really thought about before. I'm going to show you that this exposure that she had affects the response of the descendants to diet and exercise. And we think that that's because this exposure has led to heritable changes in the epigenome that permanently alter susceptibility to these. So as you heard, non-communicable diseases are on the rise. That is diseases that are not caused by bacteria, viruses, fungi, and it's now the #1 cause of death in the world. And that's pretty amazing to contemplate. The answer is we don't really know why that is, but we have some clues, and you've heard some of them earlier. What I want to talk to you about is the obesity epidemic or the obesity pandemic. And the latest statistics just out say that 39.6% of the US population are clinically obese, that is, have a body mass index greater than 30. It's disproportionately in females, which surprises my eyes, to be honest. And it's even more prevalent in the minority populations. And in African American and Hispanic females, more than 50% are obese. We care a lot about this because obesity adds a great amount of cost, both in human misery, but also to the healthcare system. And the last number is around $200 billion a year. And I like to joke at universities, imagine how many vice chancellors we could add with $200 billion a year, because that's what we seem to be good at. These costs come from are associated with increases in metabolic syndrome, type 2 diabetes, cardiovascular disease, heart attack, stroke, hypertension. 40% of all cancers occur in obese people. So there are lots of ways that these costs are passed on to society. And of course, we all know how we get obese, right? We eat too much and we exercise too little. Here's how doctors view the population, right? Any history of diet or exercise in your family. And that's true to some extent, right? You can't get fat by breathing the air. You have to actually consume calories if you want to put on weight. But there's a lot of data that says it's much more complicated. So this study came from David Allison and his colleagues. It's 6 or 7 years old now, actually 8 years old. And what they did is they looked at animal populations around the world. And they looked at really a large number, 200,000 animals from 24 different populations. Okay, and these were, yes, of course, our cats and dogs, but also wild rats, feral rats living in cities, and animals living in research colonies, monkeys, rats, mice. And what they found is they've all become obese over the last 30 years as well. So, of course, you can say that I feed my cat too much, and I probably do, but how about the feral rats? How about the rats and mice and monkeys that live in our research colonies that we give them every speck of food they have, we give them? So their diets are strictly controlled. How can they possibly be becoming obese? So something about living with people is making animals fat as well. There's another great study, and this came from an examination of the National Health and Nutrition examination study data. And I'll just summarize here what they looked over time. So in this NHANES happens every few years, and they looked over time, and they could show that between 1988 and 2006, the frequency of leisure time activity increased 47% in men and 120% in women. Right? So we're not just sitting on our butts, not doing anything. We're trying hard not to become obese. And for a given amount of caloric intake and exercise, so at the same amount of calorie intake and exercise expenditure, the BMI was 2.3 higher in 2006 than it was in 1988. So that says two things. One is this: we hear that we're lazy and not trying hard to become obese isn't true. Physical activity is increasing. And the second is this energy balance model, the diet and exercise, the calories in, calories out, cannot explain the rise in BMI. Something else is going on in addition to eating. We know there are other factors. We know that, for example, stress is associated with obesity. We know that disrupted circadian rhythms from not sleeping enough. There are genes that have some role in obesity. You've probably heard that the microbiome, the bacteria that live in your intestines, have some role in obesity. But what about prenatal experience? There's great studies from David Barker and his colleagues at the University of Southampton. Barker proposed what he called, modestly, the Barker hypothesis, or the thrifty phenotype hypothesis, that prenatal undernutrition predisposed those babies to get fat later in life. And there's a lot of data to support that idea. The Dutch hunger winter studies support that data, support that conclusion. We know that if mom smokes when she's pregnant, the babies will be born small for gestational age and will be predisposed to become obese later in life. That's from more than 35 epidemiological studies around the world in different countries, all show the same thing. Is there a role for chemicals? So there was a paper that proposed, a woman named Paula Bailey Hamilton said she couldn't lose weight after having 4 kids because she was full of toxins, and if you followed her detox diet, that you would lose weight. And that's probably not correct. But that led a guy named Jerry Heindel, then at the National Institute of Environmental Health Sciences, to write a paper about endocrine disrupting chemicals and the obesity epidemic. And this was in 2003, way before we even worked on this problem. And Jerry had the insight that many of these chemicals that are in the environment have effects on the endocrine system. Yes, there's lots of toxic pesticides that damage various things, but these endocrine disrupting chemicals work at much lower levels even than the so-called toxic levels and modify how our hormonal systems function. You probably know that weight in your body is under the control of hormones. The key ones here are, for example, leptin, which is the satiety hormone that tells your body you have enough energy. The thyroid hormone receptor, which is a member of the family of receptors that I work on, sets the basal metabolic rate, how many calories your body needs to sustain itself, basically how many calories your body burns when you're sleeping. So that is actually the biggest expenditure that your body makes in calories, is that resting metabolic rate. So if you change that, you totally change the amount of calories that you use. It's also true that the development of fat cells themselves is under the control of hormones, and 3 receptors that have the terrible name peroxisome proliferator activated receptors are the key players here. And that name is an artifact of history. These are actually fatty acid receptors, and I discovered this one when I was a postdoc at UCLA almost 30 years ago. And this receptor, PPAR gamma, is called the master regulator of fat cell development. If you express this receptor in a stem cell, that stem cell is now a pre-fat cell. And if you activate it, that pre-fat cell now becomes a fat cell. And if you activate it in a fat cell, it accumulates triglycerides from the blood. So it's a very, very important player in the whole process. You've heard the name endocrine disrupting chemicals. So endocrine disruptors, according to the Endocrine Society, are exogenous chemicals or mixtures of chemicals that interfere with any aspect of hormone action. And we have a little bit of a war with the toxicology community because they would say, and causes adverse effects. To an endocrinologist, disrupting how hormones work is adverse per se. It's adverse in and of itself. So this can be the wrong signal or the absence of the signal or the right signal at the wrong place at the wrong time. And the key point is that hormones work at very tiny concentrations. Right? So for all the women in the room, the estrogen receptor in your body is fully saturated at 1 part per billion of estradiol. That is an incredibly tiny amount. And it doesn't take much of an environmental chemical to disrupt that balance. So how are we exposed? We're exposed by persistent pollutants, the diet, food packaging, personal care products. We live in a sea of endocrine disruptors. Here's just a partial list from NIEHS. You've heard of many of these chemicals. Here's our friend 2,4-D up here, DDTs here somewhere. There's quite a lot. Some of them we know the mechanisms of action of, some of them we don't. And an important point to note here is there's probably around about 1,000 endocrine disruptors known, but that must be an understatement because there's been no systematic attempt to identify endocrine disruptors. We've learned about these by accident. Endocrine disruptors are everywhere. These are the kinds of categories, these agrochemicals, industrial flame retardants, plastics. There's quite a few of them. And we willingly expose ourselves to endocrine disruptors all the time. Most personal care products are full of endocrine disruptors, parabens, benzophenones, all kinds of chemicals like that. One argument would be that we're not really exposed. So, okay, yeah, you put the cream on your skin, but it doesn't really get inside. Well, if you believe that, I recommend this book with a very silly name, Slow Death by Rubber Duck, but it's actually a very serious book. So these guys, Rick Smith and Bruce Laurie, used themselves as guinea pigs. So they had their blood levels of a whole bunch of chemicals, phthalates, perfluorinated compounds measured, and then they sat in a room. They sprayed Scotchgard on the couch, and they sat there and they watched television. And then they had the levels measured again. And sure enough, as you might expect, the chemicals were taken up into their body, using the products as intended in just the way that you should experience them, cause the chemicals to be in your body. So the data I want to talk to you about has to do with whether endocrine disrupting chemicals are disturbing how the body functions and leading our kids to look like this. And one of the things, if you travel the world, you'll see is that in America we have lots and lots of fat kids, and you will not see that in many other countries. And that was a cartoon. We have lots and lots of obese kids here, and a really big problem is that kids who look like this very frequently turn into adults that look like this. Once a person becomes obese, it is virtually impossible to successfully maintain that weight loss. The data say that 83% of people who successfully lose a large amount of weight gain it back. And that's a very, very big problem. So how does this happen? About 15 years ago now, I and my colleagues developed what we call the obesogen hypothesis, and I'll show you a little bit about why we say that. But according to me, obesogens are chemicals that inappropriately stimulate the development of fat cells or the storage of fat into those cells, either directly by fiddling with how the fat cells work or indirectly by altering appetite, satiety, and metabolism. So was there any evidence for this even before we did this work? And the answer is yes. So these mice were generated by my friend Retha Newbold at the National Institute of Environmental Health Sciences. And the difference between these two mice is that this mouse was exposed for 5 days after birth with 5 parts per billion of a synthetic estrogen that you may have heard of called diethylstilbestrol. And then at 10 months old, this animal became morbidly obese compared to the animal that was not exposed. So that's a very, very tiny dose. We know that there are drugs that have the side effect of making people fat. So there's a kind of drugs with the terrible name thiazolidine-diones. You may have heard the name Avandia. These were diabetes drugs that make people fat. Yes, they make them insulin sensitive, but they also make them fat. And these drugs act on our friend PPR gamma. We know that there are quite a number of chemicals that you can link levels of the chemical with obesity in people. Now, of course, that doesn't prove that the chemical caused obesity, but it suggests that maybe somebody ought to study that. Because in animal models, we can directly study cause and effect. In humans, you can make associations, and you can make some inference about whether or not the effect is causal, but in animals, you can prove it without a doubt. And we know that there are bunches of chemicals that cause cells in culture to become fat cells. We take cells growing happily in a dish and treat them with these chemicals, they become fat cells. So the existence of chemicals as obesogenic chemicals was plausible even before we did the work that I'm going to tell you about. So here's our favorite obesogen, tributyl-tin. This is something we discovered completely by accident. So tributyl-tin, for a long time, was very famous as an endocrine disruptor because it was the only chemical for which there was absolutely no controversy. It exists in the environment of parts per billion, and at parts per billion, it causes this effect on mollusks. So mollusks are, the gastropod mollusks are hermaphrodites. They have both sexes, but they don't mate with themselves. They mate with other animals. And you can see what happens is a penis grows a gigantic size in the animals that have been exposed, and as you can imagine, the parts don't fit anymore. Right? So these animals become sterile. So I confess, I'm a vertebrate developmental biologist. I don't care so much about snails, except that they taste good when they're not contaminated. I was at a meeting in the south of Japan, very sleepily listening to presentation after presentation in Japanese, and one of them was in English. And Professor Tanabe got up and he said that tributyl-tin could sex reverse genetically female flounder. So a population of fish that were become 100% female, if you expose them to tributyl-tin, 30% of them became male. And that got my attention. So I called back to the lab and I said, guys, will you test which of the 48 hormone receptors that we know about that we have in the lab are activated by organotins? Thinking that it would be a sex steroid receptor, right? Because you want to change sex, you should fiddle with an estrogen or a testosterone receptor. Instead, we found that tributyl-tin activated, again, our friend, PPR gamma, and its partner, which is called RXR. So these two receptors work together as a heterodimer, and they control the development of fat cells. So there was only one way to go with these data. This was not something we were working on. It's not something that I ever contemplated studying. But here we were with this observation, this chemical activated a receptor that regulates the development of fat. So we asked the question, what happens in cell culture? And what we found is that it made cells in culture become fat cells. We found that prenatal exposing pregnant mice made the mice get fat. And we could show that this exposure reprogrammed stem cells in the body to become fat cells. So that was very interesting to us, as you can imagine. So the next thing we asked was, were these exposures heritable? And of course, we asked this question because Mike Skinner at Washington State had shown that the effects of some kind of chemical exposures were passed on many generations later. So we said, are these chemicals? Unlike Mike, we decided to use levels that are relevant to all of us. So these are the levels of chemical we use. This is a magic word, the NOEL, the No Observed Adverse Effect level in toxicology. And it's magic because that's the number that's used to set allowable human exposure. So some relationship between the NOEL and human exposure is always inferred. So we exposed these mice throughout pregnancy, and we took the babies, and we assayed some, we bred them out to the F3 generation. And if you see an effect in the first two generations, that's called a multi-generational effect. And that's distinguished from a transgenerational effect because these two generations were exposed. So if a pregnant mom is exposed to a chemical, the baby is exposed, but inside the baby are the germ cells to make the next generation, and they're exposed also. So these two generations are exposed. F3 and beyond have never been exposed to the chemical. So what we saw was that the animals weren't heavier, but they were fatter. And they had more fat cells, bigger fat cells. The brown fat didn't work normally, and they had fatty livers and lots of other problems. So I won't show you those data. They're published a long time ago. So then we repeated the experiment because we wanted to know, how did this happen? What changes did we cause in these animals? And because we wanted to know more, we went out to the F4 generation, so the great-great-grandchildren of the exposed animal. And the first thing we did is we tested all the same parameters we saw before, and we got the same results. And then we did a diet challenge to these animals. And that gave a very interesting result. So the animals were maintained on normal low-fat diet up until 19 weeks of age, and then we switched it to a slightly higher fat diet, not even double, from 13% to 21%. That's still a low-fat diet, right? Kept them on that diet for 6 weeks and then switched them back. The first thing we did is we asked, what happens when we fast the animals? Do they respond to the fasting? So normally when you fast mice, they lose weight pretty quickly. If you fast them for 4 hours, they lose some. If you fast them overnight, they lose as much as 10% of their body weight. The animals that had been exposed 4 generations before to tributyl-tin They lost a little bit less fat, but in the overnight fast, they lost a lot less fat. So these animals didn't mobilize the fat. Isn't this every dieter's lament? I don't eat and I still don't lose weight. So keep that one in mind. Second is we track body weight and body composition over time. So these are the females, and these are the males, and the 4 generations ago exposed animals are in the dotted line, and you can see there's no real difference in the weight between the males and the females. But if you look at the fat in the males, you can see that 19 weeks, they're already a little bit fatter, and that immediately when you change the diet, these animals get obese in 1 week. And then they continue to gain that weight, and when you put them back on the normal diet, they keep the weight on. So these animals are predisposed to respond differently to the diet than the control animals. That also means that body weight is not an acceptable surrogate for obesity. So a lot of times in the literature, you'll read, well, the animals didn't gain any weight, so they must not be fat. These animals didn't gain any weight, but they gained fat. So they gained fat and they lost something else. They lost lean muscle and bone mass. So we asked the question, what genes are changed in expression? And I won't bore you with going through all these pathways. Any of you who took biology, you remember memorizing these biochemical pathways. But here's the key player here. Leptin, the satiety hormone, the energy balance hormone. Leptin was elevated in the F4 male. So the messenger RNA that encodes leptin was elevated, and the circulating levels of leptin in the blood were elevated. So in the clinic, then the doctors in the room may agree with me, if you see obesity and elevated leptin levels, that means that the individual is leptin resistant. So we created animals that 4 generations after their mom's exposure had a leptin-resistant thrifty phenotype. What we think happened is we changed the way DNA is in the nucleus. So you probably don't know this, but in the nucleus of the cell, the DNA is like a bowl of spaghetti. But that spaghetti has a structure. So there are parts that like to be always next to other parts and separate from different parts. In the obesogen-exposed animals, this structure is disturbed, and that leads to heritable changes in which genes are expressed. And that this altered structure is inherited, and that leads us to get this leptin-resistant thrifty phenotype 4 generations later. And this was just published in December. So you've seen this iceberg before. How many obesogens are there? This is just the tip of the iceberg. I've shown you data for organotins. There's good data for phthalates and perfluorinated chemicals. There's really strong data for estrogens. There's really strong data for nicotine and air pollution. Who's heard Rob Lustig shouting from the rooftops about fructose and sugar? There's quite a number of other chemicals. You've heard organophosphates. Here's just a little aside I'll show you. We found in a different study that many fungicides are obesogens. Okay, here's 6 different classes of fungicides. Where are we exposed to fungicides, ladies and gentlemen? Wine, fruits and vegetables in general. So these 2 we know are obesogens in animals. These 4 activate PPR gamma, so they're for sure going to be obesogens. This one activates the PPR gamma partner, RXR. It's going to be an obesogen. And this one, we have no idea what it activates. We don't know many things yet. We don't know how many more are there. We know about 50 obesogens. Are there 500, 5,000? How many are there? We simply don't know. We don't know what the body burdens are in any of us, of any of these chemicals. They're not on the monitoring lists. We don't know what all the targets are. So we know some of the targets through which they work, but we certainly don't know all of them. We don't know much about how this prenatal exposure heritably changes the phenotype. We have some ideas in our model. Does that apply to all the other ones? So what are the implications for human health? What's the take-home message? The first is that diet and exercise by themselves do not explain the obesity pandemic. We know that because there are pharmaceutical obesogens. So even if you don't believe what I've told you about chemical obesogens, there are drugs that we take, that we prescribe for people that have the side effect of making people obese. Thiazolidine diones, the anti-diabetes drugs, all kinds of atypical antipsychotics, antidepressants make people fat. So if drugs have the side effect of making people fat, why wouldn't chemicals that target the same pathways have the same effect? That would be an unreasonable conclusion to draw. And we know quite a few of those. We know that this prenatal exposure reprograms the animals and their descendants that have been exposed to become fat. We know that there's some epigenetic changes and the changes in the three-dimensional structure of how the DNA is packed in the nucleus that leads to this predisposition. I love this cartoon. His epigenome has interacted with his butter pecan ice cream to make him fat, just exactly like my mice. This is exactly the same thing. Seriously, the existence of obesogen says we need to shift the paradigm. We need to prevent kids and adults from becoming obese rather than trying to cure people that have already become obese with an 83% failure rate. And we know how to do that. We need to reduce exposures. We need to optimize nutrition. We need to feed kids organic, contaminant-free, fresh food. I don't need to tell that message to this audience, but somehow the public health community isn't getting it. This is also one of my favorite slides. Pick your favorite government regulator, and he's saying, we do tests for the safety of pesticides in your food. It's kind of a long-term test. And it is. And we are the subjects. And you see the results, right? The results are quite clear. Chronic diseases are on the rise. People are not as healthy as they once were. I think that what to do next is quite clear. And these are the people that did this work. Raquel Chamorro-Garcia is my postdoc that's had the misfortune of doing 3 now year-long transgenerational experiments, and he's done some really great work. Bassem is a new MD PhD who did some excellent work. The NIEHS, from their little pool of funding that they have, has been wise enough to fund us. And I'm going to stop there and take any questions that you might have. No, are we doing questions? Workshop? Okay. No questions.